So I just check that you can see my PowerPoint okay. Yep.
Yep.
Brilliant. Okay. So thanks for the invitation. Today I'm going to be talking to you about how I use X-ray imaging and combine X-ray imaging and paleontology to understand the fish tree of life and vertebrate, and particularly jawed vertebrate evolution. So vertebrates are an amazingly diverse and successful modern group. And there are key features of their body plan that allow them to occupy almost any environment that you can think of. So this includes things from the land to the deep sea, and then the skies to deep underground. But there are major obstacles to understanding how this group arose. So firstly, their evolutionary roots stretch back nearly 500 million years. And secondly, lots of the most informative aspects of their anatomy are internal and therefore really hard to access. And we also have to deal with the fact that we only have fossils preserved and the issues that accompany the fossil record. So my research uses 3D X-ray imaging to try and remove these barriers and understand deep time evolution of vertebrates. Almost all living vertebrates, so over 99% of them are jawed vertebrates. And these are made up of around 1,000 sharks, rays, and chimeras up here at the top. And then the remaining vertebrates are the jawed vertebrates, the bony fishes, sorry. In contrast, there are barely more than 100 species of living jawless fishes. So these are the hagfish and lampreys. So these 100-odd species of living jawless fishes are set against the 60,000-odd species of living jawed fishes. And there's a huge morphological gap between these living jawless and living jawed fishes. So the jawless fishes, the hagfish and the lampreys, seem to have this very basic body plan, whereas jawed vertebrates have this much more diverse and disparate body plan, and they have very large sets of complex characters. These characters include jaws, 2 sets of paired appendages. So we have the pectoral fins, these are the equivalent of our arm, and our pelvic fins, the equivalent of our legs. They also have bone, teeth, and more features as well. And I'm going to talk through 3 of these features in a bit more detail. So perhaps the most famous innovation that jawed vertebrates have are the jaws and teeth. So hagfish and lamprey have keratinous plates in their mouth. These are not homologous to teeth. They've evolved separately. We've got your hagfish on the left here and a lamprey in the right. Whereas jawed vertebrates generally tend to have shedding teeth born inside their mouths on internal or external jaw bones. Another jawed vertebrate innovation is the inner ear. So jawless fishes have just this either one semicircular canal, this is in the hagfish, or they have 2 semicircular canals, this is in the lamprey. But in contrast, jawed vertebrates, including ourselves, have 3 semicircular canals. So we have an anterior, a posterior, and a horizontal semicircular canal in our inner ear. And the final innovation, and one that perhaps we don't think about as much, is the skeleton. So living jawless fishes have a skeleton that's unmineralized, made entirely of cartilage. So this picture here is the internal skeleton of a lamprey, and everything that you see there is made of cartilage. But in contrast, jawed vertebrates have mineralized skeletons made of cartilage and bone. And this includes the brain case, which is made of either mineralized cartilage or perichondral and endochondral bone. And then there's also an outer skeleton made of dermal bone, which sheaths the internal skeleton. We can try and understand the relationships between living groups of animals and draw these on a tree, on an evolutionary tree. And molecular evidence, where we look at DNA, tells us that the different vertebrate groups have these sets of relationships. So jawless fishes form one group, often called the cyclostomes, and then jawed vertebrates form another group. And within jawed vertebrates, we have these 2 groups that I mentioned earlier. So the cartilaginous fishes, the sharks, rays, and the skates, and the bony fishes, the lobed fin fishes and the ray fin fishes. And the cartilaginous fishes and bony fishes are so named for their skeletons, and we'll talk about that a little bit further on in the talk. And fossil evidence tells us that these 2 main groups of vertebrates with jaws, that is that fossil evidence tells us that these different groups of vertebrates diverged over 400 million years ago, and even as far as 500 million years ago. And this means that fossils are really incredibly important for understanding how different groups of vertebrates, particularly early vertebrates, are related to each other. Fossils are also vital for filling in this major morphological gap between the living jawless and the living jawed fishes. So we have all of these features that appear in the jawed vertebrates, but not in the jawless. And it turns out that there are two particularly important assemblages of fossils that chart the acquisition of this jawed vertebrate body plan. The first of these is an array of fishes called the Placoderms. These are a group of fishes that first evolve paired pectoral appendages to the equivalent of our arm. They're also the first animals to show dermal bone, and they're the first animals to show perichondral bone. And secondly, we have the Placoderms. And Placoderms are a really diverse group of fish that lived around 440 to 360 million years ago. They had a very diverse range of body plans, and perhaps most famous of them is this super predator here, this animal called Dunkleosteus, which would have grown to about 9 meters in length. And Placoderms are the first group of animals to evolve features such as jaws, paired pelvic appendages, teeth, and also 3 semicircular canals in the inner ear. So one of the questions that I try and answer in my research is how vertebrates became so successful and what drove different groups to become so disparate. But the backbone to understanding this is to have a really robust understanding of how different groups are related to each other. And it's fairly easy to do this in living taxa, so we can take a look at genes and look at the distribution of those genes across the different animals, but this is impossible to do for fossils. So instead, we build our understanding of relationships by observing the anatomy of each of the animals that we're interested in and breaking this down into discrete characters. And then we look at the distribution of those characters across the group of animals. So for example, we could ask whether these animals have teeth that are shed. These are absent in hagfish and lamprey. They're also absent in ostracoderms and placoderms, but they're present in sharks and then the different kinds of bony fish that we have up here. So this feature, shedding teeth, is actually one of the things that unites jawed vertebrates to the living jawed vertebrates to the exclusion of everything else. And we repeat this process of identifying characters for hundreds of taxa and hundreds of characters, and the resulting data matrix gets analyzed through this quantitative framework, and we build up a comprehensive picture of relationships. And we end up with this kind of branching pattern, with placoderms being the most closely related to living jawed vertebrates, and then ostracoderms slightly less closely related. But this is where we kind of run into our second problem. Most of the really informative aspects of anatomy are internal and therefore hard to access. So this includes things like the skeleton, but it also includes things like the brain case, which is a kind of bony box that sits inside the skull and houses the brain and sensory organs. In living animals, these features are hidden by layers of skin and muscle, and in fossils, they're also hidden by rock. So how do we get at them? Historically, people would use destructive tomography. So this is something called Solis's grinding machine. And this has been used since the early 1900s. So you basically get your fossil, you encase it in a lump of plastic, you pop it on this grinding plate, you turn this handle, and that grinds away a fraction of a millimeter. You then take your fossil off and make a pencil drawing, and you repeat that process hundreds of times until you end up with a stack of drawings. These drawings are cast into wax plates, and those plates are stuck together to create this 3D model. And this is a really amazing technique. It's been incredibly informative, but it's not ideal. Typically museum curators don't want you to take their precious fossil and grind it into dust. So today, instead of using this destructive tomography, we use CT scanning, where we use X-rays instead. So because different X-rays attenuate, because different materials attenuate X-rays differently, we can exploit this to understand the internal and external structure of an object without having to cut into it. This is exactly the same principle as getting an X-ray at a hospital. You can see your bones without having to cut through the skin. So here's an example of a CT scan through a fossil fish. Here's its eyes and some teeth, and then the white vertical line is the equivalent section that we're looking at. And you can see that different materials show up as different gray values. So inside this rock, we've got a brain case, we've got bits of the gill skeleton, we've got the jaws, and we've got the fin skeleton. So we can draw those and digitally dissect it out. And we end up with a complex 3D model of the specimen that we can 3D print and manipulate and use to look at internal and previously inaccessible anatomy. So I've told you a bit about the techniques that I'm using. So now I'm going to talk a bit about how I use them in my research. As I said, I look at early vertebrate evolution, in particular the relationships between different groups of jawed and jawless fishes. And placoderms are really key to understanding jawed vertebrate evolution. And there are lots of uncertainties around them. So in particular, the evolutionary relationships of placoderms are very unclear, both how different types of placoderms are related to each other and also how they're related to different jawed and jawless fishes. Secondly, our anatomical understanding of most of these placoderms is based on decades-old description, based on these destructive tomography techniques. These models, as I mentioned, are really informative, but they tend to have very idealized anatomy. The waxed models themselves are now too fragile for restudy. And the original specimen is a pile of dust, so we can't go back and double check those interpretations. And relatedly, there's been a tendency to view cartilaginous fishes like sharks as somehow primitive and representative of the last common ancestor of jawed vertebrates. And as a result, many kind of reconstructions of placoderm anatomy are based on this idea that they were shark-like because sharks were primitive. And this isn't particularly accurate. So let's do a bit of a deeper dive into placoderms, as they're what I'm going to be talking about next. As I said, they first appeared in the Silurian, and the animals that are classically recognized as placoderms disappear from the fossil record at the end of the Devonian. So they appear about 420 to 360 million years ago. As an assemblage, they're very diverse. So they have a huge range of body plans and ecologies, which includes this massive 9-meter long super predator. But there's also flattened animals that are very ray-like. And they were also the first animals to evolve internal fertilization and live birth. And they have a number of features that are typically associated with vertebrates. So these include jaws and teeth, although non-shedding teeth. They have paired pelvic fins, so these are the equivalent of our legs. And they also have 3 semicircular canals in the inner ear. So this is a view of the endocast, which is a sort of, here, this is a view of the endocast, which is a sort of, you infill the cranial cavity and it gives you a rough idea of what the brain would have looked like. So this is the anterior, the eyes would have been around here. And then this is the semicircular canals of the inner ear. So you've got 1, 2, and 3 of these semicircular canals. And there are many different groups of placoderms that I'm not going to go into because, frankly, no one wants to hear about words like rhinanids and acanthothoracids and ptyctodonts at this time in December because everyone's too tired. But there are 2 groups that are important to remember, and these are the antiarchs and the arthrodeers. So antiarchs tend to be dorsoventrally compressed. They have their eyes on the top of their heads, and they've got jointed pectoral fins. And then antiarchs tend to be fusiform. They look nectonic. They're nectonic predators. They look a bit more kind of classically fish-like. But relationships of these animals, as I said, is very complicated. And I've simplified this and cut some details out. But the important things to remember here is that the antiarchs tend to be resolved down at the bottom of the jawed vertebrate tree. They seem a bit more kind of similar to these ostracoderms, these jawless fishes, whereas arthrodeers tend to be resolved a little bit closer to crown gnathostomes or jawed vertebrates. Gnathostome is just another word for jawed vertebrate. And the implication of this is that arthrodeers are thought to be more similar to and more representative of the earliest relatives of living jawed fishes. So you might be wondering why we care about these arcane groups of fish and whether they're monophyletic or whether they're a group or what the relationships are. But as the closest fossil relatives to living jawed fishes, these placoderms chart the acquisition of major anatomical features like the jaws and the teeth and skeletal tissue. So without properly understanding their anatomy and their phylogenetic and their evolutionary relationships, it's very difficult to understand how key features evolved. So given current understandings of the anatomy and the relationships, what does this mean for what the earliest jawed vertebrates look like? Well, as I said, ostracoderms, these jawless fishes and antiarchs tend to be benthic, so they would have lived on the bottom at the seafloor. They would have had eyes and nostrils on the top of their head and had a generally kind of flattened body. Whereas arthrodeers and jawed vertebrates have these much more deeper bodied, they tend to have laterally placed eyes, and this indicates that they were predatory animals and they were kind of, active and towards the top of the food chain. And this suggests that there's a morphological and maybe even dietary shift or kind of predatory shift that happens around towards the top of the group. So this green arrow shows the node that unites arthrodeers and crown-jawed vertebrates, or living jawed vertebrates. Another major anatomical system that we can look at is the inner ear and the endocast. So as I said, the endocast is this bony chamber inside the skull that houses the brain and associated sensory organs. And we can use it as a proxy for brain morphology. So jawless fishes tend to have these very kind of featureless tube-like endocast, and they've got two semicircular canals. They have a wide separation between the inner ear and the rest of the brain cavity. And we see this as well in things like antiarchs. We also see it in arthrodeers, but that's very different to in living jawed vertebrates or crown gnathostomes. Living jawed vertebrates tend to have a much more well-defined endocast, and the inner ear sits very close to the endocast. So this now indicates that that morphological shift may have occurred later. So there's a set of features that are found exclusively in living fishes, in living jawed fishes, and not in the fossil group. But in order to fully understand this, we need to better resolve the relationships. And to do this, we have to target exceptionally preserved taxa that are really, really well preserved and have the potential to offer new insights into these schemes of evolutionary relationships. And an ideal candidate for this is this animal called Brindabellaspis. This is known from, it's about 410 million years old, and it's known from the early Devonian of New South Wales in Australia. And it was originally described in the 1980s. And these are really beautiful fossils. You can see here it would have had this long snout, and these two holes on the top are where its eyes would have been. And this picture is just of its head. And these were originally described in the 1980s, and it's because of its body plan, because it's quite dorsoventrally flattened and has these eyes on the top of its head, it's thought to be quite primitive. So it's thought to be quite closely related to things like antiarchs and jawless fishes. So we CT scanned two specimens. So we CT scanned the original specimen that was described in 1980, as well as a new specimen. And I worked with colleagues in Australia, China, and Sweden to do this. And these are some tomographs, reconstructed tomographs through the fossils. The host rock is a limestone. So for those of you who are familiar with CT scanning, you may know that limestone and fossil bone have got very similar chemical compositions. So that means that it's very difficult to distinguish between the two in CT scans. So that was very challenging to try and identify the difference between the bone and the host rock. But because it's in limestone, that means that it dissolves very well in acid. So several of the specimens, including this one on the far right, were acid prepared to remove all of the bone, to remove all of the limestone rock and just leave the fossil behind. So that makes it a lot easier to segment because you've just got the bone there. It's almost like you've got, a real skull with none of the soft tissue attached to it. And these were really beautiful fossils. We were able to understand lots of new things about the dermal bone, so the skeleton making up the outside of the fossil. This meant that we could look at suture patterns between different bones and identify new things that weren't apparent from just looking at the outside. So, for example, this circled plate is something called the pineal plate, which was previously unknown in this animal. But what's particularly important is that we can segment out the endocast and inner ear. So the endocast is this infilling of where the cranial cavity would have sat. And I'm showing you a picture now in dorsal view. So the big circular things up at the top of the screen, these are the nasal capsules. So these would have been what allowed the animal to smell. This bit in the middle, this kind of really bulbous bit, the optic lobes. So these would have been connected to the orbits, to the eyeball, and help the animal see. And then this big thing in this kind of bluey green color is the inner ear. And you can see the anterior, posterior, and horizontal semicircular canals. And this is a really anatomically rich region, but it's got quite a number of unsurprising features that we didn't know about before. I won't go into too much anatomical detail, but essentially it's got several features that are found in ostracoderms and jawless fishes, but are not found in living jawed fishes. So this includes a very short region of the forebrain. It's got this very kind of bulbous region further back. And then it's got this thing here called the hypothesis, which points anteriorly. And again, this is known in jawless fishes, but not so much in jawed fishes. So far, this is kind of holding up to our original interpretation of this animal as being quite primitive and early diverging and having a lot more in common with jawless fishes than with jawed fishes. But this changes when we take a look at the inner ear. And features in the inner ear are much more similar to in living jawed vertebrates than they are to jawless vertebrates. And this includes this feature here, which is called the cross commune. So this is the conjunction between the anterior and posterior canals. This is not really known in jawless fishes, but is seen in lots of jawed fishes. There's another feature here again, which is unknown in jawless fishes, but is known in lots of jawed fishes. So we've got this kind of really strange conjunction of anatomy where some things are very similar to jawless fishes and some things are very similar to jawed fishes. So what we can do is take this animal and put it into our matrix of all these different anatomical characters. And if you remember, this is our old hypothesis of relationships with Antiarchs and Brindabellaspis being quite closely related to, or being very similar to jawless fishes. And then Arthrodeers being very similar to jawed fishes, to living jawed vertebrates. But when we code this animal up into our matrix, we get a very different pattern of relationships. And we essentially flip this result on its head. So don't worry too much about the details, but just to kind of briefly talk you through it, what we end up with now in our new scheme of relationships is that Antiarchs, Antiarchs and Brindabellaspis seem much more closely related to jawed vertebrates, to living jawed fishes than we previously thought. And Arthrodeers are sort of off down here towards the bottom of the tree. So what does this mean? Well, on the face of it, it suggests that these kind of deep-bodied animals with, these flattened animals with their eyes on top of the head, it suggests that they're not actually that primitive. They're much more closely related to living jawed fishes than we previously thought. And it also suggests that Arthrodeers, this group over here, rather than being very closely related to living jawed fishes, represent this kind of independent, separate evolution of a nectonic predatory body type. And that importantly means that they're not actually very reliable. They're not very accurate for understanding patterns of evolution. We can't rely on them as this proxy for what the earliest jawed vertebrates looked like. Something that is important here is that we obviously have this very big conflict between the two results. I'm not saying that this new hypothesis is 100% right. It's probably certainly at least a little bit wrong, but it does tell us that we know far less about the origin of jawed vertebrates and the origin of things like jaws and teeth than we think we do. And probably we need to re-examine a lot of these fossils, ideally with CT scanning and other imaging techniques, and reevaluate what we think we know about them to see how accurate that understanding currently is. So let's leave the intricacies of Placoderm evolution and sort of zoom back out again to broader patterns of vertebrate evolution. And now I'm going to talk to you specifically about jawed vertebrates and the evolution of the jawed vertebrate skeleton. So here's this diagram that we saw earlier, where we've got these cartilaginous fishes, the sharks raising the scapes, and the bony fishes, the ray fins and the lobe fins. And these, if you remember, branch from a shared common ancestor over 420 million years ago. So because sharks stayed in the water and there are fewer species of them, remember there's only around 1,000 species of cartilaginous fishes compared to 60,000 odd species of bony fish. Because of this, there's often a tendency to view these cartilaginous fishes as primitive or somehow less highly evolved. So there's, we tend to think that the animal that was the last common ancestor of all the living jawed vertebrates, we think it looked a bit like a cartilaginous fish. And this seems to be held up by their anatomy. So sharks don't have bone. Their skeletons are made entirely of cartilage, but bony fish do have bone. And that indicates that bone evolved only in bony fishes. As I mentioned, cartilaginous and bony fishes are named for their skeletons. So Cartilaginous fishes, the sharks, rays, and skates, they lack dermal bone and have an internal skeleton formed of cartilage, and they've only got sort of small scales making up their outer skeleton. But in contrast, bony fishes, so these are the ray fins and lobe fins, including ourselves, have this external skeleton formed of dermal bone and an internal skeleton that's preformed in cartilage and is mostly made of endochondral bone. Although some bits of our skeleton, like our nose and ears, remain cartilage. And you can test that by, if you give your nose or your ear a wiggle, you can feel that those bits are made of cartilage. What do I mean by endochondral bone and preformed in cartilage? Well, the internal skeleton of both bony fishes and cartilaginous fishes is originally made of cartilage. And you can think about a baby's skeleton. Babies, when they're born, most of their bones aren't actually bone, they're made of cartilage. But in bony fishes, which of course includes humans, during ontogeny, that cartilage is surrounded by what we call a collar of perichondral bone, and eventually it's replaced by endochondral bone. So a good example of this is in your long bones. So for example, your arm. In a baby, that long bone, your long bones in your arm are entirely made of cartilage. And as a child grows and eventually gets to adulthood, that cartilage is all replaced by endochondral bone, apart from at the ends of the bone where it remains cartilaginous. And you have those kind of spongy caps made of cartilage at the ends of your bone. But as I said, some bits of our skeleton do remain cartilage, most notably our ears and our nose. And when we plot this onto an evolutionary tree and color code the branches according to whether or not dermal and endochondral bone are present, it looks as though the last common ancestor of jawed vertebrates lacked dermal bones. So, that animal would have been shark-like in not having big bony plates and not having endochondral bone. Another way of putting that is to say that cartilaginous fishes primitively lacked bone, and bony fish were the first to evolve bone. So in order to test this hypothesis, we can't just look at living ataxia, because living animals have all kinds of specialisms and reversals. So we need to go back and look at animals from around the time of the split between cartilaginous and bony fishes, over 400 million years ago. And this orange blob in the middle is where most of the animals that inform current hypotheses of evolutionary patterns come from. They typically tend to be from Europe and North America. But more recently, fossils with unexpected character combinations have come from new localities, and these have led to really major upheavals in the early fish tree of life. These include things from China and from Bolivia. And this, of course, motivates the fossils we study and where we look and where we form collaborations. And one intriguing area is modern-day Siberia, which is indicated by the red star up at the top. And this little fossil here is the head of a 450 million-year-old fish. It's from the depth of Siberia, and it was originally described a couple of decades ago as an early bony fish. And this is based just on its external morphology. Here are some eyes to orient you. So we're just looking at the head, specifically at the top of the skull. So up towards the top of the picture, this is where its nostrils would have been. The kind of googly eyes, which are where its eyes are. And then the back of the specimen, as you can see, this is kind of the back of its head where its neck would have been. So a few years ago, we CT scanned this specimen and used that to look at its internal and previously inaccessible anatomy. And we found that underneath the rock, this animal had a brain case that was remarkably complete and well preserved. And most importantly, despite the outside of the head looking like a bony fish with a big plate of dermal bone, the inside didn't look anything like one. In fact, the inside looked a lot more similar to placoderms or cartilaginous fishes. And this animal can tell us lots of things about the paths of blood vessels and evolution of different features and parts of the brain case, and it acts as a kind of anatomical Rosetta Stone to interpret the brain case across different groups. But more broadly, it's important because it's an example of a fossil with dermal bone in an animal that's definitely not a bony fish. When we code it into a phylogenetic analysis, we find that it falls out here, so very closely related to the last common ancestor of cartilaginous and bony fishes. And there are major implications resulting from this discovery. Most importantly, it suggests that dermal bone evolved much further down the tree than we previously thought, and was secondarily lost in cartilaginous fishes. So this means that dermal bone was present in the last common ancestor of bony and cartilaginous fishes, and was secondarily lost in sharks. So in fact, the cartilaginous skeleton of sharks and relatives is the derived state. But so far, this doesn't change our picture of endochondral bone evolution. So, this thing that develops from cartilage, that still seems to be restricted to bony fishes. But it's clear that, past narratives might be biased by where fossils have been studied from, as well as from past methodological restrictions. So the majority of our fossils, as I said, have come from Europe, and they've only been studied based on external anatomy. So it's really important to use these two new techniques and to look at previously understudied areas of the world. And another kind of intriguing and understudied location is Mongolia, which appears at the top of this paleo map of what the continent sort of looked like about 400 million years ago. Typically, when people think of Mongolian fossils, they think of dinosaurs. I'm going to move past that very quickly because the less said about dinosaurs, the better. But there's also a lot of rock from around 400 million years ago, which is the bit that we're interested in for understanding how jawed fish evolved. And although only scraps are known, these are some of the oldest jawed vertebrate fossils. So my colleague, Martin Brazeau in Imperial, has been interested in questions like this for a while, and he ran several field seasons in Western Mongolia in collaboration with the universities and research centres in Ulaanbaatar. I was lucky enough to attend one year, so here's our field crew on the right. And we flew to the capital of Ulaanbaatar, and then we basically drove west for several days. So the blue pins are where we camped each night, and then the 3 red dots in the far west of the country are where we had our field locations. And we found a really interesting locality in the far west of the country. It's somewhere called Yamak Gol. And the Mongolian Geological Survey had flagged that there were plant fossils at this location. But so we went along and we spent a few days there excavating. And this was a really amazing, if challenging location. So the strata was very steeply dipping. It was almost vertical. You can see here that the beds are sort of, almost upright. And it was also near the top of the hill at a protected location where there were snow leopards. So we had to make sure we backfilled the quarry at the end and didn't leave any trace. And the fossil was really productive, but the fossils were very, very challenging to work with. In particular, they tend to be preserved as molds on the face of the bedding plane. So what you see here is where the bone would originally have been, but it's now dissolved out. So this means that you can't dissolve the rock, because if you dissolve the rock, you dissolve any trace of where the fossil would have been. And because the rock is very hard, much harder than any remaining bone, you also can't mechanically prepare it. But eventually, after trying a few things, we discovered that CT scanning works really well. If you basically do a series of test scans and use those to identify where the fossil is, and then use rock saws to chop down the rock around the fossil, until you eventually end up with a block that's small enough to get good penetration of the X-rays through the rock. So I'm going to talk about this one specimen in particular, which is also preserved largely as a mold. So this is around 405 million years old. It's a really interesting specimen, although it doesn't look it. It looks pretty awful, but that's because, again, most of the important information is hidden inside the rock. And it turns out that this is most of the skull of a new kind of Placoderm. So one of these fossil jawed fishes that's very closely related to living jawed fishes. Down in the bottom right corner, I've given you a sort of idea. So we're looking at the top of the skull of one of these kinds of fish. And specifically, it includes the skull roof and brain case, and we can make a 3D model and segment out different bits. So the brain case is in this kind of bronze color, the skull roof is in blue, and then lots of it is molded. So this is that kind of gray color. And we can digitally dissect it, and we can take off the skull roof. We can also take off the brain case. And we end up with a model of the endochondral. So this is this cavity inside the skull that would have housed the brain. And in particular, we can see that it's got an inner ear. So here's an overview of the anatomy. So we've got the top of the skull view on the left and the bottom on the right. Again, it's partially incomplete and we're just looking at the skull. Here are its eyes again to give you a bit of a orientation. So we won't go into the anatomy, but we're going to talk about its skeleton. As this thing is a placoderm, we would expect it to have dermal bone making up its exoskeleton, which is what we see. So this is the blue on the top. But because of where it falls out in the tree, we would expect its endoskeleton, its internal skeleton, to be made of unmineralized cartilage. And as cartilage doesn't preserve in the fossil record, we would expect it, basically, when we look at a cross-section through the skeleton, we wouldn't really expect to see anything. But that's not what we see at all. So these are a series of sections, of tomographic sections through the brain case. And you can see that you've got this kind of really spongy texture inside the brain case cavity. So this isn't what we expect at all in this animal. And we can compare it to different kinds of other jawed fishes. So on the left, we have our new animal, which we name Minginia. In the middle, we have something that doesn't ossify, it doesn't mineralize its endoskeleton, it doesn't have endochondral bone. And then on the right, we have a fish that does have endochondral bone. It's a bit difficult to make out, but we get this kind of spongy texture, which looks very different to what we see in something without a mineralized skeleton, but very similar to what we see in something with a mineralized skeleton. And in fact, it looks incredibly similar to endochondral bone, this feature that's meant to be restricted to jawed fishes that we talked about before. And when we try and work out where this animal goes in our evolutionary tree, again, it comes out down here. So it comes out as very closely related to the last common ancestor of cartilaginous and ray-finned fishes. And it has really major implications for how we think bone and skeletons evolved. So rather than cartilage being primitive and endochondral bone only evolving, or in different kinds of bone only evolving in bony fishes, it looks very much like endochondral bone evolved a lot earlier than we previously thought. This again indicates that cartilaginous fishes, rather than being primitive for not having bone, are actually really specialized because they've secondarily lost it. So not only have cartilaginous fish lost dermal bone, they've also secondarily lost their endochondral bone. It's difficult to think why something would go, or why jawed fishes as a whole would go to all of the bother of evolving these different kinds of mineralized skeletons, only for cartilaginous fishes to almost immediately lose it again. But there are a few reasons why we can maybe explain this. So bone, as you can imagine, is very physiologically expensive to produce. It takes a lot of nutrients, it takes a lot of effort to mineralize and maintain that. And it also makes you a lot heavier. Bone is much, much heavier than cartilage. So it might be that cartilaginous fishes secondarily lost the ability to form bone because it just made it easier for them to swim and to spread. And this might explain why cartilaginous fishes become endemic, so seen all around the world a lot quicker than bony fishes do. So hopefully I've kind of explained to you a little bit about how fossils are not only fundamental for filling in the story of jawed vertebrate evolution, but that a rigorous and robust hypothesis of relationships is really important for understanding the evolution of different features. And we can probably only achieve this by using CT scanning to accurately understand the anatomy of different fossils. This will help us understand everything from the evolution of bone to the evolution of teeth. And CT scanning, CT-based descriptions of internal anatomy are going to be key for resolving these and other outstanding questions. So thank you very much for listening, and thank you to my funders and my collaborators for all their help.
Thank you, Sam. That was absolutely fascinating. Brilliant. Does anybody have any questions, first and foremost? Okay, well, I have one myself, which is probably going to sound really stupid, but I'm an immunologist. So does the loss of bone structure, does that potentially affect the immunobiology? I'm thinking from a human standpoint, in that humans develop quite a lot of immune cells from kind of within our bone structures. So is that likely to have affected immunobiology?
Yeah, it's possible. So there's quite a few people. So this work came out at the same time as a group of people who are working on living sharks and trying to figure out if sharks can form bone. So at the same time that we found evidence that sharks had secondarily lost the ability to form bone, they found evidence that sharks primitively did have the ability to form bone. And the reason why this is interesting is because it's very easy to repair cartilage. It's very difficult to repair bone. So if these animals, if some animals have a gene that can be switched on or off, that has really important implications for kind of medical history and that kind of thing. And that includes not only to bone repair, but also to things like immunology. So figuring out what happened and why things lost it and what the implications of losing it were, and also whether it can be switched back on is has some human and other medical implications, which I'm not going to pretend to solve, but I think it would be really cool to know if these things do have the ability to regrow bone or switch that back on.
Okay, kind of secondary to that. So where you've got the two groups, is there within the bonefish, do you get cartilaginous sections between the bone, or it does cut? So, and then essentially, is it a case then of that those cartilaginous sections have grown bigger in the kind of the evolutionary processes? That's like the cartilaginous sections have overtaken the bony sections, if you like.
Yeah, so when we're looking at fossils, even in things, so lots of things, even in bony fishes, have secondarily lost endochondral bone. So you'll CT scan something, you'll expect it to have this beautiful mineralized skeleton, and there's nothing, because it just didn't, it would have maintained cartilage throughout its life. But juveniles tend to have much more extensively cartilaginous skeletons. So if you CT scan a very small animal and it turns out that it's small because it was a juvenile rather than just because it had a small body size, then you'll see much more, you'll see much less, because much more of it would have been cartilage. And different parts, so some, for example, our jaw has got 2 ossification points. So it doesn't just grow as one bone, it grows as 2, and those kind of meet in the middle. And you can track that, the growth of those different ossifications by looking at where the gaps are.
Okay, great. So I'll start talking now. We have questions from other people. So from Tom, how can you approach segmentation of fossils in rocks of similar contrast if you have a sample which can't be acid treated?
With great difficulty. So I do, so lots of people will, if they're segmenting CT scans, they'll use a thresholding approach where you basically say anything that's this range of gray values is your thing of interest. So you just click on it and that's it. We can't do that with our data. We have to manually segment slice by slice. And you're looking for really subtle changes in contrast between the fossil and the host rock. Sometimes that's just not possible at all. So we can use different methods of tomography. So one example is using neutron tomography, because neutron tomography has a very different set of principles. So that can image things that wouldn't show up using conventional CT, but it's much lower resolution. So it's harder to get anything below about 60 micron resolution with neutron tomography. The alternative is to use phase contrast, where you're looking at, you're basically using, you're forcing additional contrast by using distance between the object at different scanning points. And the easiest way of doing that is to use a synchrotron. So I do work at the Diamond Light Source in Didcot and the European Synchrotron Radiation Facility in Grenoble. And typically we save those techniques for things that you just can't use conventional CT on because there's no contrast. And they have fantastic contrast because the X-rays are much more powerful and you take far more images. But they're also, it's a lot harder to get that data and a lot more time consuming to segment it. But yeah, mostly you just, this sort of, following up with Natasha's question on the sampling bias that affects these studies, given that cartilage isn't preserved as bone is. So if an animal doesn't preserve, if an animal only has cartilage, it's not going to show up in the fossil record. We're not going to be able to use it. And similarly, if an animal is only known from lithologies that have very similar contrast to the bone, we can't really use them. So it does introduce some kind of bias, some biases, because there are some animals that we simply can't sample unless you do destructive tomography, which museums don't really like you doing. So the sort of lucky thing is that while cartilaginous fishes have skeletons made of cartilage, some groups do mineralize that cartilage, and that does show up in the fossil record. So we do have, you know, these structures for some kinds of cartilaginous fishes, but not for others.
Oh. Okay. Any questions from anybody else? No? Okay, I think we'll call it a day there then. Thank you, Sam. Very much appreciated and a great talk.
Great, thank you very much.
Fascinating. Thank you everybody for coming. We will be back next month. I'll leave Dan to talk about that one because that's more in Dan's field.
Yes, a bit more mine. So yeah, next month we have Dr. Stuart Robertson from Loughborough University, who will be talking about FibSem and plasma FibSem, and basically the differences between them and what kind of in situ testing can be done. So that'll be quite an interesting one. So that'll be on the 12th of January, same time.
Okay.